But I want to start this morning by introducing our keynote speaker, who I've known for quite a while, Dr. Kartik Balachandran, who joins us from the University of Arkansas, where he's a professor and a graduate program director. I've known him since our days, I guess, back at Georgia Tech, where he was a PhD student when I was an undergrad there, so he's a little bit older than me. He was working with Dr. Ajay Yoganathan, who is a pioneer in the world of valve biomechanics. He finished there and went to Harvard, to the Beast Institute, worked with Kit Parker to do his postdoctoral research, and then joined the faculty at the University of Arkansas in 2012. His research has been funded by the NIH, the NSF, and the DoD, and he has a wide number of publications, all dealing with cardiovascular disease and development, and with specific interest in aortic valve disease and development. And his work has been supported by the National Institutes of Health, NSF, and DoD, as I mentioned. And we were very happy to welcome Dr. Balachandran.
All right, good morning, everyone. First, I'd like to thank Josh and FIU for inviting me here to be part of this hard day. It's very exciting. I'm excited to share some of the work that we've done over the past several years, and also to see some of the work that folks here have been doing. This amazing poster session that's been set up as I was watching it being set up this morning, and I'm looking forward to checking out what's been going on over here over the course of today. But anyway, today, so what I'm going to be sharing with you all is some of the work that we've done in trying to understand the early mechanisms of calcific aortic valve disease, specifically the role of altered metabolism and how we try to understand those events that happen early on during disease progression using non-invasive, label-free, multiphoton imaging techniques. So to begin with, just a little bit of background on our clinical motivation. So my lab in this particular area is focused on valve calcification, also known as calcific aortic valve disease or CAVD. Now in this particular type of disease, what happens is you start off with a, let's see if I can get my mouse to show up on the screen there. There we go. All right, so you start off with a healthy aortic valve, which has these compliant leaflets that open and close during each cardiac cycle, allowing for full forward flow and preventing retrograde flow. And over the course of several decades, this valve gets calcified, it forms calcific lesions, and that compromises the flow, the mechanics, and what that results in is in increased load on the heart, eventually leading to congestive heart failure. Now, 5 million Americans are diagnosed with this type of heart valve disease each year, resulting in a rising socioeconomic burden, especially in the elderly population. When we think of, in terms of treatment, really surgical replacement is the standard of care, right? So, and that usually happens when the valve has reached a stage somewhat like this, and not really all patients are operable also because of a high surgical risk and also too late or untimely diagnosis. And so what we really need is some sort of new therapeutic approach that will be able to either diagnose this disease early or allow for the treatment or reversal of some of these events that happen during the progression of calcific disease. Now, if we take a look at the pathophysiology of calcific aortic valve disease, it's complex. So starting from this schematic over here, if we take a look at what the aortic valve looks like, it has a tri-layered structure with the layer fibrosa, spongiosa, and ventricularis, with the fibrosa layer on the aortic side that is facing the outflow tract, and the ventricularis facing the ventricular side, the left ventricle. Now, these valve leaflets are populated by two types of cells. You have your valve endothelial cells that are populated on both the aortic and the ventricular sides, and you have your interstitial cells that populate the inside of the valve leaflet, the bulk of the valve leaflet. Now, As I was mentioning, the pathophysiology is complex. It's a multi-stage progressive process that is associated with a bunch of different things, including infiltration of plasma lipids, the recruitment of inflammatory cell types that include things like macrophages and T cells. I mean, all this results in the transformation of both the endothelial cells and the valve interstitial cells from a quiescent phenotype into a more activated phenotype that then interacts with these lipids and inflammatory cells to get activated, resulting in an increased number of oxidized lipoprotein deposits. And as this disease progresses further, these valve interstitial cells that were activated progressively move towards an osteogenic or bone-like phenotype. And it's during this point where these cells result in the secretion and the deposition of calcium and the ossification or the bone formation within the leaflet itself. And it's during this stage where one observes irreversible damage in terms of formation of these calcium deposits, and also it's at this stage when clinical presentation actually becomes evident. And so this is when the disease is diagnosed, and it's at this point, since it's gone beyond the point of no return to speak, surgical intervention is required. And so really, when we take a look at this progression of disease, what we want to do is not wait until this end stage here, but ask ourselves, is there a way that we can pick up the disease earlier, either from the perspective of trying to catch the disease before it progresses to that irreversible stage, or at least to understand some of the biological mechanisms that lead the disease to progress towards the point where you have these calcium deposits, and so can some intervention be done at an earlier stage? And if we take a look at the common themes that occur during the early pathogenesis of this disease, we see things like inflammation and altered metabolism happening. And so that's where my research group has decided to focus on trying to see if we can understand some of these early pro-inflammatory and altered metabolism that happens during the early stage of disease, and use that as a means to try and pick out these early it's still showing up here. Okay, good. To try and pick up these early mechanisms of disease and use that to develop therapeutic strategies. All right, so kind of putting that picture up here again, where you have your normal aortic valve that goes towards this process of inflammation, altered metabolism, eventually leading to the cells transitioning from a quiescent to an activated to an osteogenic stage, where then you have fibrosis, remodeling of the matrix, and eventual calcification. Now, if we take a look at this early stage of inflammation and altered metabolism, one of the techniques that is amenable for detecting these events in terms of altered metabolism and inflammation is label-free multiphoton microscopy, or also interchangeably known as two-photon excited fluorescent microscopy. And this particular technique has the ability to detect some of these early changes in metabolism that can alter the cell phenotype. And so what is this technique, right? So it's a little bit of background on two-photon microscopy. And most of this work is done in collaboration with two of my colleagues who are experts at this technique at the University of Arkansas. Now, in two-photon microscopy, this is where fluorophores, so things that fluoresce, are excited by two photons of half the energy but twice the wavelength. And so this is demonstrated in this schematic over here on the left, where you have two photons simultaneously hitting a fluorophore and then exciting it. And then since these fluorophores are of half the energy and twice the wavelength, this allows for the use of more visible or near-infrared region, near-infrared wavelength light to excite these fluorophores. And so this offers several advantages. Firstly, since it uses a longer wavelength, there's more depth penetration, and there's also less scattering of these excited the excitation. The longer wavelength also results in reduced phototoxicity. And one very useful thing is that this allows us to be sensitive to endogenous fluorescence. Specifically, what we are able to pick out using this technique are these metabolic cofactors, NADH, so nicotinamide dinucleotide in its reduced state, and FAD, so flavin adenine dinucleotide in its oxidized state. And over here I have just some information if you're interested in what are the excitation and emission wavelengths that are associated with picking out these particular fluorophores. Now, as you may know, these molecules, NADH and FAD, are intimately involved in a bunch of metabolic pathways in the cell. Namely things like glycolysis, so glucose catabolism, TCA cycle or citric acid cycle, and also in oxidative phosphorylation and the electron transport chain. And so they're involved in a bunch of these different processes, but what we can do is, being able to pick out NADH and FAD, we can get an intensity of each of these molecules simultaneously from your sample, and what we can do is that take that intensity and transform that into a ratio known as the optical redox ratio, which namely is the intensity of the FAD image divided by the intensity of the FAD plus the NADH image. And what this tells us, taking just basically the role that they play in glycolysis, the TCA cycle, and oxidative phosphorylation, is that this ratio will then give us a ratio of cell respiration versus cell biosynthesis. So energy consumption versus new molecules that are being synthesized, and that will also then correlate to the ratio of oxidative phosphorylation versus glucose catabolism. And so this technique has been used widely, starting off in the cancer field, and now it's getting prevalence in a whole bunch of different fields. But what we wanted to do was then take this and apply it to understanding the early mechanisms of heart valve calcification. And so in some of the early work that we did, what we have here is we took valve interstitial cells and we put them down on a flat substrate as single cells, but constraining them to different aspect ratios, trying to simulate the amount of stretch that these cells might be experiencing. And so what we have here is cells that are plated down in an aspect ratio of 1 to 3 all the way to 1 to 7, 1 to 7 being a more elongated cell, undergoing more stress within its cytoskeleton. And what we see here is that, again, this is a color image of the optical redox ratio, with red being higher and blue being a lower redox ratio. And we see that the more elongated the cell gets, the lower the optical redox ratio goes. And we're able to show that if we were to knock out the cytoskeletal stress by treating it with cytokinesin D, we don't see any change in the optical redox ratio. And so what this tells us is that this optical redox ratio was correlated with a pathological, so a slightly more stretched cell, and that was dependent on the actin cytoskeleton of that cell itself. Other groups have directly correlated these non-invasive redox ratios to the actual disease state. And so here we have work that's done from Lauren Black's group over at Tufts University showing that regions that are more calcified have a lower redox ratio, so lower multiphoton redox ratio. And this technique can also be used to pick out changes in the collagen fiber matrix as well, via second harmonic generation imaging, right? And so overall, multiphoton imaging really is a tool that is sensitive to changes in metabolism, as we've shown. And the question that we had was, is it sensitive to changes in metabolism that occur during the initiation of valve calcification? And so we asked ourselves then, can we use this technique to track the early mechanisms of disease progression, both in vitro, in simple in vitro and in vivo models? And then can we incorporate these insights into really the direction that we wanted to go towards, which was to create a physiologically relevant three-dimensional valve-on-chip microphysiological system to track some of these early events in disease progression. And so to answer these questions, what I'm going to do is present 3 research stories that we've done in our lab. The first one, where we used a simple cell monolayer model and used 2-photon excitofluorescence to track valve interstitial cell pathology, so towards disease progression. The second story is trying to take this method towards a more in vivo model, and then finally the 3D valve-on-chip work that we've done. And so the first study, can we track valve interstitial cell pathogenesis using multiphoton microscopy? And so the hypothesis that we had here was that optical redox ratio will correlate with structural, functional, and phenotypic markers during the early progression of calcific aortic valve disease, specifically only in valve interstitial cells. And so that's the cell, just to recall, which is within the valve leaflet and the one that transforms into a more osteogenic type of cell that secretes bone and results in eventual calcification. And so the study was set up as such. We had two groups. We had these cells in a quiescent media representing the healthy condition, and an osteogenic media that was meant to initiate disease in an accelerated fashion. We kept these cells for 28 days in culture, and these samples were either imaged using multiphoton microscopy, and the same samples were then taken for analysis of early and late markers for calcific valve disease progression using RT-PCR techniques. And then what we did was to correlate the imaging metrics to the PCR data to see if there was any direct correlation between the structural, functional, and known phenotypic markers for early disease progression with optical redox ratio. And so first, just to show you the model itself, and so what we have here are these cells on valve interstitial cells on cover slips over the course of 28 days. This top row here shows the quiescent healthy cells, and the second row here shows the diseased osteogenic cells. And you can appreciate the fact that these osteogenic cells, by day 14, they're forming these nodules, so the cells are coming together, clumping together, which is a very classic phenotype of this in vitro valve calcification. And then by 28 days, you see these nodules have grown quite large in size. These nodules are stained positive for alizarin red, demonstrating that they are calcific in nature. And we also show that these osteogenic nodules are proliferative and apoptotic. So again, this is just to establish that against some of these same types of studies or models that were used in the literature, that we're able to get this valve calcification occurring in vitro. Now, if we subject these to two-photon imaging, and what I'm showing you here is just a composite image of the optical redox ratio, which is taken from the individual separate NADH and FAD images. And what we see here is that it's a similar trend for both the healthy and diseased cells, where by day 14 you have a reduction in the optical redox ratio, which is much more prominent in the diseased cells. And you can see that from the data that is quantified over here as well. And then by day 28, this redox ratio goes back up again. So it's sort of going down day 14 and then going up at day 28. But the key point here is that optical redox ratio reduced significantly at the 14-day time point in these osteogenic, so the diseased cells. Now, correlating it to markers of disease, again, that we determine via RT-PCR. And so what I show you here is the optical redox ratio on the y-axis correlated with, in this first graph here, TGF-beta receptor subtype 1, and then in the second graph here, RUNX2. And so TGF-beta receptor subtype 1 is a marker for inflammation, and RUNX2 is an early marker for osteogenic differentiation. And what we see here is that there is a positive correlation with optical redox ratio. This is across all the samples and time points, and with RUNX2. Looking at more of these markers and trying to extract a little bit more information from these non-invasive multiphoton images, one thing we can do, and then this is just to point you here, we're not looking at optical redox ratio anymore, but a parameter that is known as fractal dimension. Now, since these metabolic cofactors, NADH and FAD, actually occur in the mitochondria of the cell, what we can do is then take the location of the positive expression or the positive incidence for this NADH and FAD and use that as a marker for how clustered or spread out the mitochondria of the cell is. And then what we can then do is take that clustering of the mitochondria and take the inverse of that, and we call it the fractal dimension, right? So that is essentially a metric of how clustered or spread apart the mitochondria is. And what we see here is that The fractal dimension parameter also positively correlated, but with more structural markers. So ACTA2, which is a gene that encodes for alpha smooth muscle actin, generation of stress fibers, positively correlated with this fractal dimension. And in this case here, osteocalcin, which is a late marker for osteogenic differentiation, also correlated positively with fractal dimension. And so what we can conclude from this particular set of data is that mitochondrial clustering correlated with phenotypic and structural markers. So to summarize the results that we saw from this study number 1, what we were able to say was that these multiphoton microscopy, two-photon microscopy signatures, namely optical redox ratio, decreased in time with osteogenic valve interstitial cell cultures, and also correlated with gene expression of early osteogenic and pro-inflammatory markers. Similarly, mitochondrial clustering correlated with gene expression of later osteogenic and also structural markers in these cells. So having established that we can use multiphoton microscopy to track disease progression in vitro in a simplified model, what we then wanted to ask ourselves was, can we take this to an in vivo model? Right? Can we observe altered multiphoton microscopy signatures that correlate with valve disease in vivo? And so the hypothesis here is that multiphoton microscopy, and we call it autofluorescence here, and I'll explain why in just a second. These autofluorescence ratios will be altered in response to disease progression in a mouse model of valve calcification. Now, my lab doesn't do that much of in vivo work, so what we used was a very, what I would say, a simple model for calcification, and this is a diet-based model for inducing calcification in wild-type animals. All right, so it was based off of a publication that did this in rats, and we adapted it to be done on CFP7 black mice. And so the model basically is you either feed the mice with a controlled diet, which is your open standard diet with 15 kilocalorie% fat, or a procalcific diet with an increase in vitamin D3, dicalcium phosphate, as well as cholesterol. We keep these animals for 16 weeks, and we pretty much do similar things at various time points, so it's an endpoint analysis, which is to use histology and immunohistochemistry to understand early and late markers for calcific aortic valve disease progression, and also to assess calcific aortic valve disease progression via multiphoton microscopy, right, on tissue sections at these various time points. All right, so just to show you the model itself. All right, so, and then again, this is at the 16-week time point, to show you what the phenotype of this particular diet-based model for calcification is. And so, for those of you more familiar with valve calcification, you'll find that this does not look exactly like how the human presentation of valve disease it should be. But what we see is positive oil red O and alizarin red staining more in the commissures. So to orient you, this is a transverse section of the aorta. You can see the 3 valve leaflets over here. And if you zoom into the commissures are where the valve leaflets attach to the aortic root, and you see that positive oil red O staining, positive alizarin red staining at the commissures. So again, this is not exactly valve calcification, but at least there is some positive calcification somewhere. And so then maybe we can take a look at the multiphoton imaging metrics at those same regions and see if there is some correlation. And so we then also further validated it using the classic markers for valve calcification, and we were able to show that again, control in the first column, and then the one that was fed the procalcific diet in the second column, that you have positive alpha SMA staining, positive Ki67, so cell proliferation, positive RUNX2, and those same commissures, osteopontin, as well as TGF-beta type 1, so showing your classic activated cells, proliferating cells, markers for early osteogenic differentiation and pro-inflammatory cytokines. And then now what we wanted to do, and then again, taking sequential, so adjacent sections, and subjecting them to multiphoton microscopy. And so here's where it gets slightly complicated, because we have extracellular matrix proteins and a whole bunch of other things in tissues that are not there in cells. And so if we take a look and then if you focus your attention here on the top left, this table here, you have your excitation wavelengths here on the top row and your emission wavelengths here in this first column. Previously, with just cells, we could excite them and be very sure that we were only getting emission or fluorescence from the NADH and FAD. But for tissue sections, we have sort of bleed through from things like collagen and calcium that might be in that tissue that show up at the same wavelengths, all right? And so now we can't call this an optical redox ratio anymore, so we just call it an autofluorescence ratio at the same wavelength. So specifically, a TPEF, which is 2-photon excited fluorescence, 755 to 860 autofluorescence ratio. It's essentially the same thing, we're just not calling it optical redox ratio anymore. And so what we see here is that, just like what we observed in the in vitro study with the cell monolayer, we see a reduction in this autofluorescence ratio at the commissure, all right? So you can see slightly more blue compared to the oranges that we see in the control, as well as the earlier diseased group time points. And we see that in the 16-week animals that were fed the procalcific diet, there's a reduction in this autofluorescence ratio. And then we quantified this for the leaflet, the commissure, and the root region of the valve, and we see the same trends. So at the 16-week time point for the animals that were fed the procalcific diet, there is a reduction in this autofluorescence ratio. And if we were to correlate this, now we see an inverse correlation between this autofluorescence ratio and markers such as RUNX2 and proliferation. Right? So RUNX2 for osteogenic differentiation and Ki67 for proliferation. And so this, we didn't really take this study too far. We also did look at other wavelength excitation and emission wavelength combinations, but since this didn't really demonstrate a phenotype that was similar to the actual valve calcification phenotype, we kind of ended the story here, and we were able to say that mice that were fed a procalcific diet showed increased lipid and calcium deposition, osteogenic differentiation, inflammation, and proliferation at 16 weeks, and at the same time showed a reduced 755 to 860 multiphoton autofluorescence ratio at 16 weeks that correlated with these same markers for calcific disease. So it was good in the sense that this imaging technique is able to at least pinpoint the same regions which had increased expression of these disease markers. So that was great for us. But now what we really wanted to do, and what we were much more interested in, was to take this towards a valve-on-chip model. So we wanted to build a more physiologically relevant benchtop model to study these early mechanisms, and then these mechanisms specifically as it relates to altered metabolism. So if we take a look at the current models for studying aortic valve disease, they're quite varied. You have, for example, some models that were developed by us where you have a stretchable membrane with a well that holds a 3D hydrogel construct that mimics the valve leaflet. You also have other techniques where these hydrogels are constrained by springs, and these springs are attached to actuators that allow them to expand and contract, just like how a valve leaflet does in vivo. You also have models where cells are micropatterned to mimic the shape and size and length that they are in vivo. And then you also have some other models which incorporate things like hydrogels, fluid flow, three-layered structures to try and mimic the tri-layered structure of a valve leaflet. If you're interested, so I have the QR code for a review paper that we have published recently, summarizing some of these in vitro models that exist out there. And then if you take a look at in vivo models, there are a variety of in vivo models that exist, either using some diet intervention like what I showed you in our work, or some gene knockout models that knock out specific genes that are known to potentiate disease in these, to potentiate valve disease. Now what we wanted to do was then really focus on the in vitro, but try to develop a model that is more advanced than some of the models that exist out there. And so if we take a look at our design parameters, if we take a look at what we really need to do to build a valve on chip that is more physiologically relevant, that mimics the in vivo environment, we need, if we take a look at what this valve leaflet actually looks like, it's pretty complex. Like I mentioned to you, you have your aortic side, the ventricular side. The valve leaflet is made up of 3 layers, right? So you have your fibrosa layer, which is near the aortic side, that is comprised of these collagen fibers. You have a spongiosa layer, the middle layer, that is made up of proteoglycans, right? So, or glycosaminoglycans. And then you have your ventricularis layer, the bottom layer, that is comprised of elastin fibers. This entire tissue is being stretched as it opens and closes, and it's also being subjected to 2 different kinds of shear stresses. On the aortic side, you have oscillatory shear stress, and on the ventricular side, you have laminar shear stresses, which are more pulsatile in nature as the heart is pumping and when the valve opens and closes. So taking all this, we summarize some of the considerations for engineering a valve on chip, which is it needs to include the cell types that comprise the valve, so endothelial and interstitial cells, and maybe immune cells, since the immune cells are an important aspect of disease initiation and progression. We definitely want a multi-layered tissue structure, and so we need a complex ECM hydrogel, which contains things like collagen, glycosaminoglycans, and elastin. And we also need a way to impose dynamic mechanical forces. So this would be the stretching, the fluid shear stress, etc. And such a model does not exist, all right? So a model that recapitulates the multi-layered structure, BAEC, BIC, so endothelial interstitial cell co-culture, and dynamic mechanical stimulation. So that doesn't really exist. And so what we wanted to do was address this particular research gap. And so the first model that we developed looked something like this, right? Schematically, we had a 2-layer model, so the fibrosa, so that is that collagen-rich layer that faces the aortic side, and the spongiosa, which is the more fluid glycosaminoglycan layer. So we constructed these layers using, so the fibrosa layer using methacrylated collagen, and the spongiosa layer using a combination of methacrylated collagen and glycosaminoglycan, so chondroitin sulfate and hyaluronic acid. All right, and so here's just some information on the composition of these different components that we use to construct these hydrogels. Right, so we, to effectively study disease, it's also important to mimic both the healthy and the disease composition of the extracellular matrix of the valve leaflet. And so we did that by increasing or simulating the enrichment of glycosaminoglycans that exists during early disease progression, as shown here in the table. So we constructed a disease hydrogel that had 4 times the amount of glycosaminoglycan compared to the healthy hydrogel. And then, since these were methacrylated, we were able to use UV cross-linking techniques to polymerize this gel and create a hydrogel for growing these cells. Now we wanted to be able to then take this hydrogel and put it in a structure that will allow for imposition of mechanical forces. And to do that, what we did was to use a stretchable PDMS or polydimethyl siloxane silicone polymer housing. And so what we have here is 2 clamps holding a PDMS sheet, and within this PDMS sheet we had our housing that the hydrogel would be placed in, right, and bonded to, so to allow for effective strain transfer from this moving sheet to the hydrogel itself. So we assemble these 2 layers, UV cross-link them in this hydrogel, and you can see here, this is a picture of what the actual setup looks like. You have a linear actuator. We have 2 of these samples here, and then this is a side view showing the gel and the culture media within the housing here. So then we were able to then take this sample, and then we were to ask our question. So the hypothesis here is that multiphoton microscopy autofluorescence ratios will be altered in response to disease progression in this 3D valve on chip microphysiological model, and they will correlate with protein markers for altered metabolism. And so the groups that we had were either a healthy version of this hydrogel or a diseased version of this hydrogel, both by a careful manipulation of the hydrogel composition, as well as the appropriate quiescent versus osteogenic media composition. So since we were interested in the earliest of mechanisms that lead towards valve disease progression, our initial study was to take this and subject it to 48 hours of 10% cyclic strain within this valve, this flexible PDMS housing. And then we did pretty much the same technique. We would then first assess the sample via multiphoton imaging, so image the samples and then take those same samples. And this time, we would isolate proteins for an analysis via proteomics. All right. So just a quick view of what these samples look like. So here we see health, images of healthy disease, and we see that the samples are alive, the cells are alive, and then they express stress fibers within these hydrogels, much like what we expect them to do in vitro culture. We wanted to phenotype these cells. Healthy hydrogel cells in the healthy samples express vimentin and are not positive for alpha SMA, and the reverse is true for the disease samples where they are highly positive for alpha SMA, showing that the disease samples exist the cells within the disease samples exist in a highly activated state. Now the multiphoton imaging. So if you recall, I told you when I was talking about the second story, once you have ECM proteins in there, things get a little gnarly in terms of using multiphoton imaging to tease out NADH and FAD, right? It becomes a little bit more difficult to discriminate between what is the contribution from the extracellular matrix proteins and what is the contribution from the mitochondria of the cell. Now to overcome this, what we can also do with this multiphoton microscopy technique is to image what is known as fluorescence lifetime, right? So fluorescence lifetime specifically of NADH. And so this is using the, so exciting NADH and counting the lifetime of the emission of the fluorophores. And the fluorescence lifetime of NADH is highly sensitive to either its free or protein-bound states, right? So where the free NADH is mostly found during a glycolytic state, and protein-bound state is more found during an oxidative phosphorylative state, right? So then what we can do then is count these photons and plot a decay curve like this, right? And then using these coefficients and then using plotting a bi-exponential fit to this decay curve, we can then extract these two constants here, A1 for the first time constant and A2 for the second time constant. And what we can do then is take this ratio of A1 by A2, where the first time constant A1 represents the relative contribution of free NADH, so more representative of glycolysis, and A2 is a constant that correlates more with the relative concentration of protein-bound NADH, so more towards oxidative phosphorylation. And so that's what we show here. So again, these are our fluorescence lifetime images. The first column here shows in a color map the actual lifetime of the samples, the different regions of the sample, and then on the right column here is a generated image of this A1 by A2 ratio, right? And so as the ratio goes higher, that means it's becoming more glycolytic. If the ratio goes lower, that means you have more oxidative phosphorylation. And so what we see here is that there's a significant decrease in this A1 by A2 ratio, meaning the diseased samples had reduced glycolysis, increased oxidative phosphorylation taking place, right? And so we were able to tease this out and not be affected by some of these other fluorophores contribution from the ECM. Then on to proteomics, right? So again, the next few slides are going to be pretty busy with a lot of proteomic data, so I'm going to walk through that systematically. And so our proteomic data revealed the following, right? So it revealed that there were 26 unique proteins present in the healthy samples, 170 unique proteins that were present in the disease samples. Now, if we take a look at these proteins that were commonly expressed between healthy and disease samples, so here 1,424 different proteins. And after assessing the fold change in expression of these 1424 proteins we found that there were 14 significantly upregulated proteins, so that's shown here, and 12 significantly downregulated proteins. Right? We can express this, these same 14 upregulated and 12 downregulated proteins in the form of a heat map, just for easier visualization. Now, some of these significantly upregulated proteins included things like lipid oxidation, cell matrix interactions, and the downregulated proteins included things like cholesterol biosynthesis, endopeptidase inhibitors. Now what we wanted to do was just dig deeper into some of these upregulated and downregulated proteins and to see if any of these correlated with the fluorescence lifetime metrics that we obtained using multiphoton imaging. And so just a few slides on some of that data. Right, so if we take a look at some of these proteins that were up and down regulated, so the first one here, FDPS, which is farnesyl diphosphate synthase, and so this is an enzyme that's involved in cholesterol biosynthesis, right? So and it was downregulated in the diseased valve chip samples, and what we saw was that it was positively correlated with this A1 by A2 ratio. Remember, this A1 A2 ratio, we can call it a ratio of representative of decreased glycolysis, and we saw that there was a positive correlation between this FDPS enzyme and the A1 by A2 ratio. Here's another one, EEA1, so this is early endosome antigen, which is a marker for the early endosome, and it can signify increased lipid uptake. And we saw that there was an inverse correlation between this EEA1 protein and A1 by A2 ratio obtained from multiphoton imaging. And so based on this, we're able to hypothesize, some pathways for further, deeper study, and we can say that, perhaps in the disease samples, when you have decreased glycolysis, that results in eventually decreased cholesterol biosynthesis, and at the same time increased expression of EEA1 protein that results in increased cholesterol uptake. And what does this mean for disease? We don't know. We're still trying to figure it out and dig deeper. Just a couple more things, just to show you what we have. We also found that this A1 by A2 ratio correlated positively with, over here, heat shock protein. Correlated negatively with heat shock protein HSPH1, and then positively with PSMD1 protein. Again, these two proteins are involved in things like the folding and unfolding of proteins and the aggregation of proteins and stress in the endoplasmic reticulum. So clearly something is happening in terms of altered cholesterol biosynthesis, changed protein folding, and increased endoplasmic reticular stress that we are interested in digging deeper and going further. Now, always when you develop an in vitro model like this, we want to make sure that we are building something that looks like in vivo, like the human tissue, right? And so that's what we are doing here. And so to characterize the validity of our valve chip samples against human samples, we compared the significantly altered proteins, what we're calling deregulated here, in disease samples with proteins that are significantly altered in diseased human aortic valve samples that we were able to obtain. And among the significantly altered proteins in the diseased samples as compared to healthy, we found that 5 of the altered proteins were common between our valve-on-chip samples and human aortic heart valves. And again, these were also markers of things like complement regulation, proteasomal degradation, the early endosome, cell ECM interactions, and then these are all clear hallmarks of early disease progression. And then for further analysis, we performed a gene ontology analysis for these human samples, and we found that our samples had deregulated fatty acid and cholesterol metabolism, similar to what was observed in the diseased human valves. And just one final slide on the proteomics. We also performed PCA analysis and gene ontology on the positively and negatively loaded proteins, and we found something quite interesting actually. So what we have in these PCA plot is the following. So if you take a look, again, we have limited access to human disease samples and also healthy human valve samples. And so here you have our single calcified valve way out here, and over here we have our healthy valve on chip samples, and then here we have the clustering of our disease valve on chip and a normal or undiseased human valve. And so what we see here is that what we're calling disease valve on chip doesn't really look like a full-on calcified human heart valve, but it looks more like a non-calcified but aged human valve. All right, so somewhere in an intermediate disease process, and of course it's not exactly like a healthy sample at all. And so we're still trying to work further, tweak the ECM composition, tweak some of the things that we're doing to our model to see if we can mimic some, different stages of the actual human disease, and we're working more towards that as well. And so to summarize this aspect here, what we were able to say is that multiphoton microscopy can detect the early metabolic changes in valve, endothelial, and interstitial cells. In our disease samples, we're calling them as cells that are in an early disease initiation stage based on the fact that they were marked by an acute adaptive response to stress, and our PC, our physical component analysis, showed that they were more like an aged human heart valve that was not fully calcified yet, and so we're calling it an early disease initiation state. These disease valve and chip samples showed altered protein homeostasis, reduced cholesterol biosynthesis, and a preference towards oxidative phosphorylation over glycolysis. And just very quickly, so what we're working on now is, so like I showed you, we have a two-layered model with a layer of endothelial cells and a fibrosa and a spongiosa layer. What we're trying to do now is to improve it to look more like an actual valve leaflet, so 2 layers of endothelial cells and 3 layers of extracellular matrix, just like in the valve leaflet, imposing both flow on the top and the bottom side. And so towards that, what we've done is to develop something like this, where we have a porous membrane holding all these gels together and a 2-layered valve on chip, where you have flow on the bottom, flow on the top, mimicking your oscillatory flow, pulse type flow, and then this is something that we hope to take further and build on this and try and tease out some of these disease mechanisms. And then here's just a video of this thing in action here, and this is what it looks like. All right, so to end, I thank you again for the invitation and then for your attention here. I'd like to thank all my students, postdocs, especially Dr. Ishta Tandon, shown here. This entire talk was basically her PhD work. Our funding from the NIH, NSF, and the AHA for this particular project, our collaborators at the Proteomics Research Center over at the University of Arkansas, imaging collaborators, Kyle Quinn and Tim O'Leary. And I'll be happy to take any questions, but before that, I want to give a quick plug about my department, since I'm also the graduate coordinator. So we're recruiting master's and PhD students. Again, I know I don't have too much time right now, but I just wanted to let you all know. So we have a bunch of different programs, so 3 master's programs, non-thesis, thesis, and an interesting one that is partnered with our business school, the Walton College of Business, where we have students really working with business students and trying to develop their ideas from the research lab towards a startup. And so that's the whole focus of the program. And then of course we have our traditional PhD program as well. And a whole bunch of different research areas: cancer, cardiovascular, neuro, regenerative medicine, tissue engineering, orthopedics. And finally, I think this will be the most important slide for everyone, is we have a bunch of fellowships that are offered. We have this distinguished doctoral fellowship and a doctoral academy fellowship, and basically what this gives you is additional money beyond your GRA stipend and a tuition waiver. So for the DDF fellowship, we have students who, after adding on the stipend and the tuition waiver, making up to $50K per year, and I'll be here this whole day, so if any of you folks are interested in finding out more, please find me and then we can talk more about this, all right? So again, thank you for your time, and I'll be happy to take any questions.
Balachandran.
So thank you for your talk.
Push the button.
So thank you for your talk. This was very informative for me. I was really curious to see what for some of the I guess underlying model assumptions behind the design of that organ on chip model.
Yeah. So were you interested in anything specific in terms of like what gel we use, what aspect are you more interested in?
I think I guess just like in general. I guess
Yeah.
maybe a more key assumption, like a more fundamental.
Yeah, so I think that's a good question. It's also a very open-ended question here. So I think what we really wanted to do was mimic the structure, right? So that was, that was something that we placed a lot of importance on. So we wanted to mimic a healthy structure, disease structure, and a healthy structure and disease structure. And so that's where we had the tweaking of the extracellular matrix composition of the collagen, the glycosides, proteoglycans, etc. We also wanted to impose mechanical stretch at the first stage of the model. And so the assumption there was that LP leaflet undergoes 10% cyclic strain, and so that's why we imposed that 10% over there. We wanted both cell types, and so again, endothelial cells, interstitial cells, and so that was kind of the basis for that as well. Hope that answered the question in terms of, yeah.
Testing. Okay, awesome. Thanks again for the talk. That was awesome to watch. I had two questions. The first one being, you talked a lot about the calcification in terms of the hallmark of the disease. I was wondering if in your mouse models or in your human data, did you notice any differences between the metabolism between the left and right coronary cusps?
Good question. We didn't really pay close attention to that, but I think, at least from what I recall of the data for the mouse data, we did not, I mean, again, that mouse model didn't really show calcification, like what calcification really is, right? So we saw, these, I can go back at the beginning, actually look at it right now, and see if there's anything interesting there.
Yeah, but I guess you're already kind of touching upon my second question, which was in your mouse models, even though you didn't see, this hallmark calcific nodule by alizarin red positive staining on those leaflets themselves, did you see any other aspects of pathological remodeling, leaflet thickening or excess collagen position, and did that correlate with any of your markers?
Yeah, that's a good question, and I didn't show it here, but it is in the paper that was published for this data. And so you can see here, I mean, it seems like all the commissures had positive alizarin red staining, but then to that question, we did see altered collagen, at least from second harmonic imaging of the leaflet itself, and so that did correlate with, in terms of what we would expect to see in the disease, so leaflet thickening and altered structure of the collagen microarchitecture. So we did see that. We did see increased expression of RUNX2 on the leaflet itself, and so we do see some of those things. We just didn't observe positive alizarin red staining on the leaflet itself. Yeah, and it seems, back to your first question, we didn't really notice anything specific in the non-coronary side of the leaflet that stood out. Yeah.
Yeah. Hello, and thank you for your presentation. That was amazing, and it's great to learn all these things that are going on right now. The organ on chip is what my question is about. Everything's kind of moving away from the animal models towards this direction, and your valve on chip seems to be kind of pushing forward that. How far away do you think you are within your valve on chip to having a workable model to actually replace the animal models that are currently not as applicable to the human condition?
Yeah, that's a good question. I mean, I'm biased, so I would say they're ready for replacing animal models. I don't know if John will agree with that. No, I think I think it's going to be a slow transition. If you're talking about drug development, the pharma industry still heavily relies on animal models, right? And that's not going to change anytime soon, I think. But maybe there might be a slow progression towards where these benchtop valve on chip models can at least work together in collaboration with these animal models to inform either disease mechanisms or in the drug design and testing realm.
Can I ask a follow-up on that? Because, well, have you tried to see, can you start with something that looks like the healthy leaflet, and instead of trying to mimic the disease leaflet, drive it toward the disease?
Yeah, we have actually, and it's again nothing that's published or we presented on it, but we can. So just by tweaking the media or adding inflammatory cells, we can create some sort of disease progression in the healthy sample as well. Yeah, for sure.
Hi, thank you for that talk. I was wondering if you saw any sex-related differences in your mice or their human samples.
Yeah. So in the valve-on-chip samples, we weren't able to discriminate between any sex differences, but in the mouse study, yes, we did see differences. In terms of the male mice had much more accelerated progression towards calcification compared to the female mice that we had. Yeah, and again, the paper has some of that data discriminated.
Other questions?
This one here.
Sorry.
So other than for calcific disease, I was also wondering if the multiphoton microscopy can also be used to catch on to other diseases as well.
Yeah, for sure. I think any disease that has changes in metabolism, you can use this technique. And so, like I mentioned at the start, cancer, which is a disease that has huge metabolic changes within the cell. That's where this whole multiphoton microscopy kind of got started. Wound healing as well is another huge aspect of where one can use multiphoton imaging to track changes in wound healing, fibrosis, and those kinds of things. So yeah, definitely is widely applicable for whenever there is some metabolic change happening in the cell. This technique is applicable.
All right, let's thank Dr. Balachandran.
Thank you.
We're now going to move on to the getting ready for the poster presentation. So we'll start at 10:30. So we have a little bit of time to kind of get set up. So poster